Assalamualaikum and hi everybody. For MIC662 TASK3 will be discussed on the article titled Acacitine attenuates Streptococcus suis by simultaneously targeting psilocin and inflammation. This study will be reviewed by Norajiba Yusra and Noraida under the supervised Dr. Shafiq Azmi. The article examines the potential of acacitine, a chemical derived from the traditional Chinese herbs, to be used as a therapeutic agent for treating drug-resistant Streptococcus suis infections. The research focused on the inhibitory effects of acacitine on the toxic psilocin and its mechanism of action. According to the findings, acacitine might be used as an alternate therapy for highly pathogenic and drug-resistant SS2 infection. The objectives of the studies are to find and assess acacitine as the chemicals that target SLY and S-Swiss virulence factors and regulates the host inflammatory response. Second, to investigate acacitine as the alternative therapeutic option for treating S-Swiss infections, especially those produced by the virulent and drug-resistant strains. The significance of the study discussed that acacitine was discovered to suppress SLY's hemolytic action and to decrease the inflammatory response. It is also no harmful effects on the cells at specific doses indicates that it might be used for therapeutic. This shows that acacitine might be utilized as the alternative therapeutic for highly pathogenic and drug-resistant SS2 infections. The first methodology is the hemolysis assays. The researchers conduct the hemolysis assays to assess the inhibitory effects of acacitine on the hemolytic activities of psilocin, a virulence factors of SS2. Different concentrations of the acacitines were tested to determine the effectiveness in reducing hemolysis. Second, the growth curves. The growth of SS2 bacteria is the presence of acacitine was monitored using growth curves. This allows the researchers to evaluate the impacts of acacitine on the bacterial growth and viability. Western blot assays were performed to analyze the expression of inflammatory markers and the phosphorylations of kinases involved in the mitogen-activated protein kinase pathway. This helped determine the effects of the acacitine on the host inflammatory response. Lastly, molecular dynamics simulations were conducted to visualize the binding process between acacitine and the active center of SLY. This provided insights into the mechanism of action of acacitine in inhibiting SLY and reducing its virulence.
Moving on to the results, this research found that acacitine inhibits the hemolytic activity of psilocin from at least 4 microgram per ml. At the same time, it reduced the oligomerization of psilocin. Oligomerization is the interaction of protein subunit to form larger and functional complexes for bacterial virulence, and acacitine successfully reduced the SLY oligomerization, which simultaneously diminished its hemolytic activity. However, the drug alone is unable to kill SS2 and still depend on host immunization for pathogen clearance. Next, acacitine protects cells from infection damage by downregulating the MAPK pathway. Mitogen-activated protein kinase or MAPK pathway is a signaling cascade that regulates the inflammatory response and involves activated kinases B38, ERK, and JNK. Upon infection, activated MAPK pathway may trigger severe inflammatory response that can cause diseases like septic shock and meningitis by Streptococcus suis. Acacitine successfully slows down inflammatory response while showing non-cytotoxicity effect on healthy cells. Additionally, as higher concentration of acacitine shows protective effect to host cells, it is important to find an appropriate dosage to avoid cytotoxicity effect. The LDH assay shows that concentration below 64 microgram per ml is non-toxic to the cell, and 8 to 32 microgram per ml is sufficient to provide protective effect. The authors continue to suggest 16 microgram per ml as appropriate dose with almost no dead cells, as shown in figure F. Lastly, this study viewed the binding of acacitine with the active center of SLY located between the domain 2 and domain 4 by hydrophobic interaction. The binding free energy of Val475 and GLY476 near the acacitine destabilizes the hydrophobic core of SLY toxin during the binding process of acacitine and SLY's active center. The destabilization hinders oligomerization and consequently inhibit hemolytic activity and reduce the SS2 virulence. In conclusion, acacitin has the potential to be developed as a novel therapeutic agent to treat drug-resistant Streptococcus suis. This study shows that acacitin are able to inhibit the virulent factor of Streptococcus suis type 2 while causing non-cytotoxicity effect and reducing the harm to cells by downregulating the inflammatory response. Therefore, further clinical investigation for acacitin to combat Streptococcus suis infection are necessary. And lastly, this is the reference, and that's all from us. Thank you.